The following content is provided by Google Scholar. Introduction: Major depression is one of the most common psychiatric disorders in the Western world and is the leading cause of disability, as measured by years lived with a disability. In the year 2000 it was the fourth leading contributor to the global burden of disease, and by the year 2020 is projected to be second only to ischemic heart disease in the amount of disability experienced by sufferers. World Health Organization, 2008. Major depression is a heterogeneous disorder recognized as having a complex and multifactorial etiology originating from the interaction between environmental and genetic factors. As depicted in Figure 1, these lead to a range of disturbances including neurotransmitter imbalances associated with lowered serotonin, dopamine, and noradrenaline, and increased glutamate activity, hypothalamus, pituitary, adrenal, HPA, disturbances typically characterized by increased HPA activity and glucocorticoid resistance, dysregulated inflammatory pathways as evidenced by increased levels of pro-inflammatory cytokines, acute phase proteins and kynurenine pathway metabolites, increased oxidative and neuroprotective damage associated with reduced antioxidant defenses, neuroprogression resulting in neurodegeneration, apoptosis, reduced neurogenesis and neuronal plasticity, and mitochondrial disturbances in the form of increased damage to mitochondria and mitochondrial DNA, and lowered ADP production. Manji et al., 2001, Moss et al., 2009 C., Raison and Miller, 2011, Leonard and Moss, 2012. It is these multiple pathways that make the effective treatment of depression difficult, with current therapies achieving far from optimal remission rates of between 20 to 40%. Warden et al., 2007. This has motivated a search for alternative pharmacological, psychological, environmental, and even nutraceutical treatment options to enhance current treatment outcomes. Given the multifactorial nature of depression, an enhancement of treatment efficacy is likely to occur from therapies that target multiple mechanisms. This is likely to be achieved by combining therapies and/or utilizing treatments with multiple mechanisms of action. Curcumin, derived from the spice turmeric, is a potential natural substance that may at least partly fulfill this criterion. As demonstrated in Figure 2, curcumin has been shown to have a large array of molecular targets of action, which may prove to be beneficial for the treatment and prevention of depression and other diseases. Agarwal and Sung, 2009, Gupta et al., 2012. This has already led to two published brief reviews highlighting the potential antidepressant effects of curcumin, Kulkarni et al., 2009, Kulkarni and Dhir, 2010. Interest in the benefits of curcumin has increased dramatically over the past decade, with a number of clinical studies currently underway investigating its efficacy for treating and preventing a range of diseases including certain cancers. MD Anderson Cancer Center, 2012, Alzheimer's disease, Martins and Arjuna natural extracts, 2012, and musculoskeletal conditions, University of California, 2012. In addition, at least one unpublished study has been completed investigating the antidepressant effects of curcumin, Government Medical College, Bhavnagar et al., 2012, although the outcomes of this study have not yet been released. The research completed to date on the multiple effects of curcumin is reviewed in this paper, with a specific emphasis on the biological systems that are compromised in depression. The antidepressant effects of curcumin in animal models of depression are summarized, and its effect on neurotransmitters, HPA disturbances, inflammation, oxidative stress, mitochondria, neuroprogression and intestinal hyperpermeability, all of which are disturbed in depression, are detailed. Animal behavioral models of depression. Despite the fact that no animal model is able to incorporate all aspects of depressive symptoms in humans, the forced swimming test, FST, is a reliable and valid tool for screening the antidepressant effects of drugs. Petit Demoliere et al., 2005. Animals, typically rats and mice, are subjected to two trials where they are forced to swim in an inescapable cylinder filled with water. The time animals spend immobile in the second trial provides a measure of despair, and has been shown to be reduced by antidepressant drugs. Petit Demoliere et al., 2005. The tail suspension test, TST, is another animal model of depression that has good reliability and predictive validity. Cryan et al., 2005. In the TST, animals are subjected to the short-term, inescapable stress of being suspended by their tail, with time spent in an immobile posture providing a measure of depressive behavior. Various antidepressant medications have been shown to reverse immobility and to promote escape-related behavior. Steru et al., 1985. Antidepressant effects of curcumin in animal models. The antidepressant effects of curcumin have been evaluated through the FST and TST in over a dozen studies in the past decade. These have consistently demonstrated that both acute, Kulkarni et al., 2008, Wang et al., 2008, Gilatra and Dingra, 2000, Sanmukani et al., 2011, administration of curcumin to rats and mice reduce immobility time. For example, both in the acute models of FST and TST, and the chronic model of FST with a water wheel, curcumin had significant antidepressant-like activity compared with a vehicle control. The effects of curcumin were similar to those of fluoxetine and imipramine, although when administered in combination, antidepressant effects were not enhanced. Sanmukani et al., 2011. Similar results have been found in other animal models of depression, with curcumin having similar antidepressant efficacy to fluoxetine. Wang et al., 2008, Li et al., 2009. Bilateral olfactory bulbectomy in rats causes changes in behavior, and in the endocrine, immune, and neurotransmitter systems, that lead to many characteristics seen in patients with major depression. Bulbectomy is believed to cause major dysfunction in the cortical, hippocampal, amygdala circuit similar to that seen in people suffering from major depression. Song and Leonard, 2005. Chronic, but not acute administration of antidepressants corrects most of the changes evoked by bulbectomy. Song and Leonard, 2005. A study assessing the effects of curcumin demonstrated that its chronic administration was also able to reduce immobility time and reverse the behavioral abnormalities induced by bulbectomy. Su et al., 2005a. By depleting monoamines and increasing oxidative stress, reserpine is a drug that increases pain and depressive symptoms. An investigation into the protective effects of curcumin on the reserpine-induced pain, depression dyad in rats demonstrated that curcumin was effective in ameliorating a number of behavioral changes induced by reserpine. Aurora et al., 2011. Finally, depressive-like behaviors characterized by a decrease in sugar consumption and an increase in immobility time were induced by 3 weeks of corticosterone injections in rats. Concurrent treatment with curcumin reduced depressive-like behaviors, demonstrated by a 46% increase in sucrose consumption and a 57% reduction in immobility time compared with rats receiving corticosterone injections alone. Huang et al., 2011. Based on the studies reviewed, in animal models of depression, oral and intraperitoneal curcumin appear to have potent antidepressant properties with effects similar to antidepressants such as the serotonin reuptake inhibitor fluoxetine and the tricyclic imipramine. Depression and immunoinflammation. Interest in the association between major depression and inflammation was triggered by studies demonstrating increased levels of inflammatory mediators in depression. This led to the development of inflammatory models of depression and prompted initial comparisons between the symptoms of depression and sickness behaviors such as anorexia, soporific effects, reduction of locomotor activity and exploration, andonia and cognitive disturbances. Raison et al., 2006, Dancer et al., 2011. These were later confirmed to be distinct conditions, with sickness behavior an adaptive, acute state, whereas depression is a disabling, progressive disorder. Moss et al., 2012a. 3 recent meta-analyses have all confirmed that depression is associated with elevated levels of pro-inflammatory cytokines and other inflammatory mediators. Howren et al., 2009, concluded that depression in clinic and community samples was associated with high levels of C-reactive protein, CRP, interleukin-1, IL-1, and interleukin-6, IL-6. In a meta-analysis by Dowlady et al., 2010, levels of tumor necrosis factor alpha, TNF-alpha, and IL-6 were significantly higher in depressed patients than controls. Finally, a meta-analysis by Liu et al., 2012, demonstrated that the blood levels of soluble interleukin-2 receptors, SIL2R, TNF-alpha and IL-6 were all significantly higher in patients with major depressive disorder than in controls. There is also evidence that major depression is characterized by a Th1-like cell-mediated response, as evidenced by increased production of interferon gamma, IFN gamma, increased IFN gamma/IL-4 ratios and increased neopterin levels. Moss et al., 1994, Myant et al., 2005. Further support for the relationship between depression and inflammation is provided by a meta-analysis on the anti-inflammatory effects of antidepressant medications, which concluded that antidepressants reduced levels of cytokines IL-1 beta and possibly IL-6, but not TNF-alpha, Hanstat et al., 2011. The N-methyl dispartate, NMDA, receptor antagonist ketamine, which has rapid antidepressant effects in treatment-resistant patients with major depressive disorder, also has anti-inflammatory effects, Loya et al., 2011, Matthew et al., 2012. While increased inflammation is not found in all patients with depression, it is argued that its effects may be relevant to a subset of patients, Raison and Miller, 2011. Researchers have also become increasingly interested in the impact of inflammation on kynurenine pathway metabolites which have both neurotoxic and neuroprotective qualities, Dancer et al., 2011, Moss et al., 2011 c. Immunoinflammatory effects of curcumin on inflammatory pathways. Extensive research using a wide range of in vitro models has indicated that curcumin can reduce the inflammatory response by regulating the production of a range of inflammatory molecules, Basnet and Skako-Basnet, 2011, Shrivastava et al., 2011. Research performed in animals also provides strong evidence for the beneficial effects of curcumin in various diseases associated with inflammation including inflammatory bowel disease, obesity, cardiovascular disease, and certain cancers, Gupta et al., 2012. Evidence of anti-inflammatory effects in human studies is accumulating and has so far been promising. For example, in a clinical trial on osteoarthritis patients, the administration of a patented form of curcumin, Mediver-registered, significantly reduced a number of inflammatory markers, including IL-1 beta, IL-6, and the erythrocyte sedimentation rate, ESR, Belcaro et al., 2010. In another randomized clinical trial, curcumin, BCM95 registered, given to patients with rheumatoid arthritis significantly reduced the ESR to levels similar to that obtained from the nonsteroidal anti-inflammatory drug, diclofenac sodium, Chundran and Goyal, 2012. Further evidence of the anti-inflammatory effects of curcumin was demonstrated in a randomized, double-blind, placebo-controlled study on patients with overt type 2 diabetic nephropathy where curcumin administration reduced serum levels of TGF-beta and IL-8 and urinary IL-8, Kajetahi et al., 2011. Finally, in another study on patients with type 2 diabetes, curcumin lowered levels of IL-6 and TNF-alpha significantly more than placebo, Ushrani et al., 2008. COX-2 selective inhibitors are a form of nonsteroidal anti-inflammatory drug, NSAID, that directly targets COX-2, an enzyme responsible for the production of prostaglandin E2, PGE2. PGE2 is a pro-inflammatory chemical messenger involved in pain, fever, and swelling. In normal conditions, COX-2 is not expressed in most cells, but elevated levels are found during inflammation, which, in turn, stimulates the production of PGE2. As a cofactor, COX-2 is associated with the increased expression of ETO, Basu et al., 2006, Lee et al., 2009, Cesario et al., 2011, and with inflammatory cytokines, Abbasi et al., 2012. Studies in the area of depression have provided evidence of increased PGE2 production and COX-2 expression in depressed people. For example, increased concentrations of PGE2 in the saliva of patients with major depressive disorder were identified, Ohishi et al., 1988. Moreover, increased levels of PGE2 were reported both in the CSF and serum of depressed patients, Linoilla et al., 1983, Calabrese et al., 1986. Increased expression of the genes encoding for COX-2 were also found in the peripheral blood cells of patients with recurrent depression, Galecki et al., 2012. The antidepressants fluoxetine and amitriptyline also inhibit cytokine-induced PGE2 production by inflammatory cells, Yaron et al., 1999. Recent interest in the potential of COX-2 inhibitors to augment antidepressant therapies also shows promise, with a number of positive findings from animal models of depression and from preliminary human clinical trials, Muller and Swartz, 2008. In a 6-week double-blind, placebo-controlled trial, depressed patients treated with a combination of fluoxetine and the COX-2 inhibitor seloxib experienced greater improvement in mood compared with those treated with a combination of fluoxetine and placebo, Okhonzade et al., 2009. In a double-blind, add-on study using the noradrenergic reuptake inhibitor reboxetine as the antidepressant, Moeller et al., 2006, obtained a similar outcome. The combination of reboxetine and seloxib led to significantly greater improvements compared with the reboxetine alone treated group. Also, in an open-label pilot study, the mixed COX-1/COX-2 inhibitor acetyl salicylic acid accelerated the antidepressant effect of fluoxetine and increased the response rate in depressed non-responders to fluoxetine, Mendelwiz et al., 2006. More recently, seloxib enhanced the efficacy of the serotonergic antidepressant sertraline in 40 depressed patients. Patients in the seloxib and sertraline group experienced a significantly greater response, 95%, and remission, 35%, rate compared with those receiving placebo and sertraline, 50% response and 5% remission, Abbasi et al., 2012. An investigation into the biological activities of curcumin has demonstrated that it can downregulate COX-2 expression and PGE2 synthesis, Plummer et al., 1999, Hong et al., 2004, Moriyuki et al., 2010, Lee et al., 2011, therefore providing an alternative natural option as a COX-2 inhibitor. While further human clinical studies are required in this area, the promising antidepressant findings of pharmaceutical COX-2 inhibitors provides another potential mechanism of action for the antidepressant effect of curcumin. Effects of curcumin on immune pathways. EDO is an enzyme expressed in multiple cell types including macrophages, dendritic cells, astrocytes, and microglia and is strongly activated by the pro-inflammatory cytokine IFN-gamma and to a lesser extent TNF-alpha, IL-1 and IL-6. EDO can also be induced by lipopolysaccharides, LPSs, Chen and Guillemot, 2009, Stone et al., 2012. As shown in Figure 3, EDO is important in depression as it reduces serotonin synthesis by catabolizing tryptophan, the primary amino acid precursor of serotonin, into kynurenine pathway metabolites. The role of serotonin in depression is already well recognized, however, accumulating research shows that a number of kynurenine pathway metabolites can impact on several mechanisms associated with depression, Moss et al., 2007, 2011c. For example, excess levels of the NMDA agonist and kynurenine pathway metabolite quinolinic acid, quin, are excitotoxic, leading to degenerative changes in the CNS. It can increase the generation of free radicals and induce lipid peroxidation. The damaging effects of quin can also contribute to mitochondrial degeneration, and in experimental models quin has anxiogenic effects, Stone et al., 2012. Excess quin levels were found in depression, Raison et al., 2010, Steiner et al., 2011, and in neurological conditions such as Alzheimer's disease, Parkinson's disease, and multiple sclerosis, Saderi et al., 2009, Kinks's et al., 2010. Another kynurenine pathway metabolite and NMDA antagonist, kynurenic acid, kyna, is considered neuroprotective and inversely regulates dopaminergic activity. Its balance with quin is important as it is considered protective against the excitotoxicity of quin, Myant, 2012. Kyna levels were found to be low in depression, Myant et al., 2007, Moss et al., 2011b, and, more recently, were observed to be particularly low in patients with somatization, Moss et al., 2011b, Moss and Reef, 2012. Kyna levels were also associated with an increased risk of depression in people undergoing IFN-alpha treatment for hepatitis C virus, Wichers et al., 2005. Research into the effects of curcumin on EDO expression is only preliminary. However, studies identified to date have been positive. One study demonstrated that curcumin significantly inhibited the expression and activity of EDO in IFN-gamma-stimulated mouse bone marrow dendritic cells, Zhang et al., 2009. In a study on cancer cells induced by IFN-gamma, curcumin was also reported to inhibit the expression of EDO, Zhang et al., 2008. Finally, in an in vitro and in vivo investigation, curcumin delivered to mice significantly attenuated LPS-induced increases in EDO expression in bone marrow-derived dendritic cells, Young et al., 2010. Further evidence of curcumin's immunomodulating effects are demonstrated by its ability to lower Th17 autoimmune responses, Sia et al., 2009, Konakazabai et al., 2012, which have recently been shown to be upregulated in major depression, Chen et al., 2011. Curcumin also decreases the expression of Th1 cytokines, e.g. IFN-gamma, TNF-alpha, and increases the expression of Th2 cytokines, e.g. IL-4 and IL-10, Zhang et al., 2006, Bearswill et al., 2010. This may prove to be beneficial for the treatment of depression given consistent findings of increased Th1-like cell-mediated responses in major depression, Moss et al., 1994, Myant et al., 2005, Moss, 2011. Several studies have also investigated the influence of curcumin on the LPS-induced immune response. One such study revealed that the systemic administration of LPS to rabbits increased core temperature and hypothalamic levels of glutamate and hydroxyl radicals along with increased plasma levels of TNF-alpha, IL-1 beta, and IL-6. Pre-treatment with curcumin 1 hour before an intravenous dose of LPS significantly reduced the LPS-induced overproduction of circulating TNF-alpha, IL-1 beta and IL-6, and brain glutamate, PGE2 and hydroxyl radicals, Huang et al., 2008. Depression and neurotransmitters. The monoamine deficiency theory is the most recognized and investigated causative model of major depression. It is postulated as the primary mechanism of action of popular pharmaceutical antidepressants, such as selective serotonin reuptake inhibitors, SSRIs, and serotonin noradrenaline reuptake inhibitors, SNRIs, and is thought to increase the availability of monoamines such as serotonin, noradrenaline, i.e., norepinephrine, and, possibly, dopamine. According to this theory, the underlying pathophysiological basis of depression is a depletion of these neurotransmitters in the CNS, Maladek et al., 2007. Serotonin is the most extensively studied neurotransmitter in depression, with evidence of its deficiency emerging from studies using tryptophan depletion, which reduces central serotonin synthesis, Toker et al., 2010. Studies demonstrating abnormalities in serotonin receptors in depressed patients, Carr and Lucky, 2011, research showing increased availability of monoamine oxidase, an enzyme that metabolizes serotonin and other monoamines in the brain of depressed people, Meyer et al., 2006. And evidence of abnormalities in the expression of the enzyme tryptophan hydroxylase, which is involved in serotonin synthesis, Mathis et al., 2010. The monoaminergic theory has prompted research into the potential of curcumin in altering neurotransmitters such as serotonin, dopamine, and noradrenaline. A selection of these studies is reviewed. Effects of curcumin on serotonin, dopamine, and noradrenaline. Both animal and in vitro studies conducted over the past decade have now confirmed that curcumin is able to influence levels of serotonin, dopamine and noradrenaline in the CNS. For example, in a chronic unpredictable mild stress, CUMS, model with rats, curcumin reduced serum corticosterone levels and attenuated CUMS-induced reductions of serotonin, Lee et al., 2009. Curcumin administered to mice for 21 days prior to the FST was also shown to markedly attenuate FST-induced decreases in concentration of serotonin, the serotonin metabolite 5-hydroxyindoleacetic acid, noradrenaline, and dopamine, as well as increases in serotonin turnover, Sia et al., 2007. Similar results were found following the acute administration of curcumin to mice 1 hour prior to the FST. Curcumin dose-dependently increased serotonin, and at higher doses increased dopamine levels. However, no changes in noradrenaline levels were found. The researchers demonstrated that curcumin was able to inhibit the monoamine oxidase enzymes, both MAO-A and, at higher doses, MAO-B, and when co-administered with piperine, a bioavailability enhancing agent. The effects of curcumin on pharmacological, biochemical, and neurochemical activities were potentiated, Kulkarni et al., 2008. Similar findings were obtained when rats were subjected to chronic unpredictable stress. Curcumin treatment for 21 days significantly reversed the chronic unpredictable stress-induced behavioral, increase in immobility period, biochemical, increase in monoamine oxidase activity, and neurochemical, depletion of brain monoamine levels, except noradrenaline, alterations. The combination of piperine with curcumin again showed significant potentiation of its anti-immobility, neurotransmitter enhancing, serotonin and dopamine, and MAOA effects compared with curcumin alone, Butani et al., 2009. In an animal study on the effects of curcumin on serotonin, 5-HT, receptors, the antidepressant-like effects of curcumin in the FST were related to the serotonergic system, possibly due to an interaction with 5-HT1A/1B and 5-HT2C receptors, Wang et al., 2008. Further evidence of the monoamine effects of curcumin has been demonstrated in rats submitted to a bilateral olfactory bulbectomy. The administration of curcumin completely reversed induced reductions in serotonin and noradrenaline in the hippocampus and frontal cortex, Su et al., 2005a. Curcumin also reversed risperin-induced reductions in dopamine, noradrenaline, and serotonin in rats, Aurora et al., 2011. Pretreatment with curcumin reversed haloperidol-induced reductions of dopamine, noradrenaline, and serotonin in rats, Bishnoi et al., 2008. In a follow-up study, the co-administration of piperine significantly enhanced the effect of curcumin on these monoamines, Bishnoi et al., 2011. Collectively, these animal studies provide strong support for the monoaminergic effects of curcumin. Curcumin enhances levels of serotonin and dopamine and, to a lesser extent, noradrenaline, following exposure to acute and chronic stress. The studies reviewed in this section indicate that curcumin has an inhibitory effect on monoamine oxidase and influences 5-HT1A/1B and 5-HT2C receptors, all of which play a role in depression. Effects of curcumin on glutamate. Glutamate is the major excitatory synaptic neurotransmitter in the CNS and plays a vital role in the regulation of synaptic plasticity, learning and memory, McEntee and Crook, 1993. Increasing research has demonstrated altered levels of glutamate in plasma, cerebrospinal fluid, CSF, and various areas of the brain in people with depression. An increasing number of reports also suggest potential antidepressant effects of antiglutamatergic agents, such as ketamine, amantadine, and riluzole, McNally et al., 2008, Gao and Bao, 2011. Curcumin inhibited the release of glutamate evoked by exposing synaptosomes to the K-end channel blocker 4-aminopyridine, 4-AP, Lin et al., 2011. Because the inhibitory effect of curcumin on glutamate release was completely abolished by the antidepressant oxidative, it was concluded that these two substances may use a common intracellular mechanism to inhibit glutamate release from rat prefrontal cortex nerve terminals. When the effects of curcumin and fluoxetine were compared, curcumin had significantly greater inhibitory effects on glutamate release than fluoxetine, Lin et al., 2011. Depression and HPA disturbances. The HPA axis plays a significant role in the body's stress response and is initiated by the secretion of corticotrophin-releasing hormone, CRH, and arginine vasopressin, AVP, from the hypothalamus. This, in turn, activates the secretion of adrenocorticotropic hormone, ACTH, from the pituitary gland, which then stimulates the secretion of glucocorticoids, cortisol in humans and corticosterone in rodents, from the adrenal cortex. These glucocorticoids impact on their receptors and are involved in the negative feedback control of CRH and AVP release from the hypothalamus, Zunsain et al., 2011. Abnormalities in the activity of the HPA axis have long been observed in major depression, peri-anti-enlightenment, 2008. This has been characterized by heightened cortisol secretion in patients presenting with melancholic depression, and reduced levels in people presenting with atypical depression, Gold and Krausos, 2002. Increasing research has also demonstrated that depression is associated with a hypersecretion of CRH and impairment in the responsiveness to glucocorticoids, a phenomenon known as glucocorticoid resistance, peri-anti-enlightenment, 2008. Depression is also associated with an increased size and activity of the pituitary and adrenal glands, Nemiroff et al., 1992. Effects of curcumin on HPA disturbances. A few studies have examined the effect of curcumin on regulating HPA disturbances, namely its effect on moderating the hypersecretion of corticosterone and subsequent neuroprotection. For example, rats subjected to 3 weeks of corticosterone injections exhibited depressive-like behaviors, as demonstrated by a significant decrease in sucrose consumption and an increase in immobility time in the FST. These behaviors were significantly reduced in those rats treated concurrently with curcumin, Huang et al., 2011. In a study on rats exposed to 4 weeks of chronic unpredictable mild stress, curcumin was able to moderate increases in serum corticosterone at levels similar to those obtained by fluoxetine, Li et al., 2009. In a final study, exposure of corticosterone to rat neurons decreased mRNA levels for 3 serotonin receptor subtypes, 5-HT1A, 5-HT2A and 5-HT4. Pretreatment with curcumin 1 hour prior to corticosterone exposure reversed this effect on 5-HT1A and 5-HT4 receptors, but not for the 5-HT2A receptor. Moreover, curcumin significantly reduced neuronal loss, indicating that it could protect cells from corticosterone-induced toxicity. Curcumin also inhibited corticosterone-induced morphological changes such as increases in soma size, dendritic branching and dendritic spine density, and elevated synaptophysin expression in cortical neurons. The antidepressant fluoxetine also provided a protective effect against the toxicity of corticosterone and reversed corticosterone-induced downregulation of mRNA levels for 3 receptors, 5-HT1A, 5-HT2A, and 5-HT4, Su et al., 2011. In sum, these studies highlight the potential protective effects of curcumin on stress-induced cortisol production. HPA imbalances in depression may prove to be at least partly ameliorated by curcumin, although further studies are required. Depression and neuroprogression. Neurogenesis is the process by which new neurons are formed from populations of neural stem or progenitor cells residing in discrete regions of the CNS, Abdi-Pranoto et al., 2008. Recent research into neuroanatomy and neurochemistry has increasingly demonstrated that major depression is associated with impaired neurogenesis, neuronal plasticity and subsequent neurodegeneration. This is indicated by stress-induced alterations to the number and shape of neurons and glia in the brain regions of depressed patients, Drummond, 2009. Depression is also associated with a decrease in proliferation of neural stem cells, Adrian, 2012. Neuronal plasticity is influenced by a range of neurotrophic factors, such as brain-derived neurotrophic factor, BDNF, which is the most abundant and widely distributed neurotrophin in the CNS, Martinowich and Liu, 2008. BDNF plays a role in regulating a large array of functions including neuronal survival, growth, and proliferation, and levels usually are low in people suffering from major depression, Drummond, 2009, Lee and Kim, 2010. Chronic administration of several classes of antidepressants, including monoamine oxidase inhibitors, SSRIs, tricyclic agents, and SNRIs, increases BDNF levels, Drummond and Monteja, 2006, Sen et al., 2008. The detrimental effects of early life and chronic stress on BDNF, and evidence of increased levels of BDNF following physical exercise and electroconvulsive therapy, provide further support for a role of BDNF in the pathogenesis of depression, Martinowich et al., 2007, Nagahara and Tujinsky, 2011. Neuroprotective effects of curcumin. Preliminary investigations into the neuroprotective effect of curcumin have provided evidence of its ability to enhance BDNF and attenuate stress-induced neurodegeneration. Repeated corticosterone injections were shown to significantly decrease BDNF levels in the hippocampus and frontal cortex of rats, which curcumin was able to significantly ameliorate, Huang et al., 2011. In another study, rats exposed to a range of daily stressors over a 20-day period were administered either 3 doses of curcumin orally or the antidepressant imipramine. Curcumin administration at higher doses reversed the stress-induced decrease in hippocampal neurogenesis in stressed rats at levels similar to imipramine treatment. Curcumin also reversed the stress-induced decrease in BDNF and 5-HT1A mRNA levels across all hippocampal subfields. These effects were similar to those obtained from imipramine, Su et al., 2007. Further evidence for the neuroprotective effects of curcumin was demonstrated in a recent study where pretreatment with curcumin reversed the effect of corticosterone-induced neuronal changes in rats, Su et al., 2011. Curcumin also attenuated quinolinide-induced excitotoxicity on primary cultures of human neurons, Brady et al., 2011. Depression and mitochondrial disturbances. Mitochondria are intracellular organelles found in most eukaryotic cells which generate most of the cell's supply of adenosine triphosphate, ADP. They are also involved in a range of other processes, such as signaling, cellular differentiation and cell death, as well as the control of the cell cycle and cell growth. McBride et al., 2006. Because of the brain's significant energy demands, high concentrations of mitochondria are found in this region, making the brain susceptible to reductions in aerobic metabolism. Pichanek and Neustadt, 2007. Malfunction in the biochemical cascade and damage to the mitochondrial electron transport chain can lead to mitochondrial dysfunction or disease, which has been implicated in a range of neuropsychiatric disorders such as depression, bipolar disorder, and schizophrenia. Rezin et al., 2009, Gardner and Bowles, 2011. In depression, mitochondrial disturbances such as deletions of mitochondrial DNA, Gardner and Bowles, 2008a, Shao et al., 2008, and lower activities of respiratory chain enzymes and ADP production, Gardner et al., 2003, have been found. Gardner and Bowles, 2008a, 2008b, also showed that in depressed individuals with somatic complaints, the majority had lowered ADP production rates in biopsied muscles. Further support for mitochondrial dysfunction in depression is demonstrated by increased rates of depression in patients with mitochondrial disorders. For example, the lifetime diagnosis of depression in patients with mitochondrial disorders was 54%. Fatal et al., 2007. Greater rates of depression were also detected in adolescents with mitochondrial disorders. Cohen et al., 2009. It has been argued that because depression is associated with increased oxidative and nitrosative stress, increased inflammatory pathways and lowered antioxidant defenses, this may be a major cause of mitochondrial dysfunction in major depression. Gardner and Bowles, 2011, Moss et al., 2012 c. Kynurenine pathway metabolites such kynurenic acid, 3-hydroxykynurenine, and 3-hydroxyanthranilic acid, which are dysregulated in major depression, can also impair mitochondrial ADP production. Barron et al., 2003. Effects of curcumin on mitochondrial function. Altering the pathogenic cascade associated with mitochondrial dysfunction in major depression may be a potential avenue to enhance treatment outcomes in depression. Curcumin, with its numerous molecular effects, may be a useful option with a number of studies demonstrating protective effects on mitochondria. Curcumin was able to attenuate oxidative damage in rat cortical neurons by reducing intracellular production of reactive oxygen species, ROS, and protecting mitochondria from oxidative damage. Zhu et al., 2004. Treatment with curcumin also lowered at Purdue University Library TSS on August 29, 2014. Jop.sagepub.com. Downloaded from 1518 Journal of Psychopharmacology 26, 12. Aluminium-induced oxidative stress and mitochondrial dysfunction in the rat brain, likely by several mechanisms such as activation of Hem oxygenase 1, maintenance of glutathione metabolism, or by scavenging ROS. Sood et al., 2011. Curcumin and its analogues were also able to inhibit ROS-induced lipid peroxidation and protein damage in mitochondria. Wei et al., 2006. Nuclear factor erythroid 2-related factor 2, NRF2, which mediates neuroprotection against mitochondrial complex ion 2 inhibitors, is also activated by curcumin. Greco and Fiscum, 2010, Zhang et al., 2011. Depression and nitric oxide. Nitric oxide, NO, is an intercellular messenger in the brain, synthesized from the amino acid L-arginine by the enzymatic action of nitric oxide synthase, NOS. 3 NOS isoforms are known to exist in mammals: neuronal NOS, NNOS, inducible NOS, INOS, and endothelial NOS, ENOS. Alderton et al., 2001. NO plays an important role in various physiological and pathological processes, and there has been increasing interest in its role in depression and other mental health conditions. MacLeod et al., 2001, Pinto et al., 2008, Dhir and Kulkarni, 2011. According to Dhir and Kulkarni, 2011, support for a role of NO in the pathogenesis of major depression is provided by studies showing that levels of plasma NO and its metabolites are elevated in suicidal and depressed patients. Suzuki et al., 2001, Lee et al., 2006. Evidence that decreasing or blocking the synthesis of NO can induce antidepressant-like effects. Joka and Gomariz, 2006. research demonstrating that no modulates the production of neurotransmitters such as noradrenaline, serotonin, and dopamine, Tyr and Kulkarni, 2011, and evidence of lower levels of NNOS in the locus coeruleus of people suffering from major depression than in normal controls, Carroll was et al., 2004. Further indirect evidence for no involvement in major depression is derived from the characterization of circulating antibodies against no epitopes. Highly reactive substances such as no tyrosine, no tryptophan, and no arginine are formed by nitration reactions, and increased IgM antibody levels against these noaducts have been found in depressed patients, Moss et al., 2008b, 2011d. These results suggest that increased NO/or activation of nitration reactions and consequent increases in nitrate-containing proteins may be involved in the pathophysiology of depression, Moss et al., 2011a. Effects of curcumin on nitric oxide. Several studies have now confirmed that curcumin can regulate levels of nitric oxide. Immobilization-induced restraint stress lasting 6 hours significantly increased plasma nitrite levels in mice, which was attenuated by intraperitoneal delivery of curcumin. This effect was enhanced when curcumin was jointly delivered with aminoguanidine, an INOS inhibitor, thereby suggesting curcumin's possible inhibitory effect on INOS, Gilatra and Dingra, 2010. In cultured human neurons, quin induced NNOS activity and consequently increased nitrite levels. Curcumin dose-dependently decreased NNOS activity in human neurons, Brady et al., 2010. Further evidence of curcumin's nitric oxide inhibitory effect was demonstrated in a study on rats subjected to 72 hours of sleep deprivation. This led to increases in nitrite levels, along with several markers of oxidative damage. Intraperitoneal treatment with curcumin for 5 days had anxiolytic effects and restored nitrite levels. This effect was, however, abolished by pre-treatment with L-arginine, a precursor to nitric oxide, but only with the lower dose of curcumin of 10 mg per kilogram. The researchers concluded that nitric oxide modulation is involved in the protective effect of curcumin in ameliorating sleep deprivation-induced behavioral alterations and oxidative damage, Kumar and Singh, 2008. Depression and oxidative stress. In a review paper, Moss et al., 2011a, confirmed that major depression was accompanied by a decreased antioxidant status and by the induction of oxidative and nitrosative pathways. This was supported by studies showing significantly reduced plasma concentrations of important antioxidants such as vitamin C, Kanzode et al., 2003, vitamin E, Moss et al., 2000, Owen et al., 2005, and coenzyme Q10, Moss et al., 2009b, in depressed patients. Depression was also accompanied by a lowered total antioxidant status, Sarandal et al., 2007, Kyomurku et al., 2009, and reduced antioxidant enzyme activity such as glutathione peroxidase, Moss et al., 2011e. Such deficiencies can impair protection against ROS, causing damage to fatty acids, proteins, and DNA, Moss et al., 2011a. In fact, increased levels of malondialdehyde, a measure of lipid peroxidation, were found in depressed patients, Ojkan et al., 2004, Sarandal et al., 2007, Wei et al., 2009, as were increased levels of 8-hydroxy-2-deoxyguanosine, 8-ODG, a measure of oxidative damage to DNA, for Lenza and Miller, 2006, Moss et al., 2009a. Further support for an association between oxidative stress and depression is provided by a few studies that demonstrate antioxidant properties of antidepressant medications. However, not all studies have uniformly identified reductions in oxidative markers following antidepressant medications, necessitating further research in this area, NG et al., 2008. Clinical studies into the efficacy of antioxidant therapies to alleviate depressive symptoms are lacking, although N-acetylcysteine, NAC, a powerful antioxidant, was found to be useful for depressive episodes in bipolar disorder, Magalhaes et al., 2011. Zinc, which serves as a strong antioxidant, also has evidence of antidepressant activity, Zukzik et al., 2011. Antioxidant effects of curcumin. Investigations into the pharmacodynamics of curcumin have consistently demonstrated it to be a potent antioxidant, Menon and Sithier, 2007, AK and Gulson, 2008, at least 10 times more active as an antioxidant than vitamin E, AK and gulsin, 2008. In relation to a selection of the oxidative markers associated with depression, curcumin is able to lower levels of malondialdehyde, RI-ETAL, 2010, and 8-ODG, Shi and Lin, 1993, RI-ETAL, 2010, NUYUK-ETAL, 2011, and increase the activity of the antioxidant enzymes superoxide dismutase and glutathione peroxidase, MONICUNTUN-ETAL, 2011, NUYUK-ETAL, 2011. Curcumin also attenuated increased markers of oxidative stress in rats undergoing chronic stress, BODI-ETAL, 2011. In recent studies, curcumin also upregulated NRF2, a transcription factor that induces the expression of various genes including those that encode for several antioxidant enzymes, Yang-ETAL, 2009, Scopagnini-ETAL, 2011. At Purdue University Library TSS on August 29, 2014 jop.sagepub.com downloaded from Lopresti ETAL 1519 Depression and intestinal hyperpermeability. The intestinal epithelium is the largest mucosal surface in the human body and provides an interface between the external environment and the host. Fasano and Shea Donahue, 2005. Normally, the intestinal epithelium provides a semi-permeable wall which allows nutrients to be absorbed while preventing larger, potentially toxic, antigenic, or pathogenic molecules or organisms from passing into the bloodstream. However, the permeability of the intestinal epithelium can become compromised, leading to a condition known as intestinal hyperpermeability or leaky gut. This results in an increased diffusion of antigenic food molecules and translocation of bacteria from the gut into extra-intestinal sites, which can then trigger an immune response. Miller, 1997. LPSs are large molecules found in the outer membrane of gram-negative bacteria and, during a state of intestinal hyperpermeability, are known to translocate into the systemic circulation in greater quantities. Moss et al., 2008a. Once the LPSs are translocated into the blood, an inflammatory process is generated and is associated with increased neuroinflammation and secretion of pro-inflammatory cytokines, such as IL-1 beta, IL-6 and TNF-alpha. Dunn, 2006, Cheen et al., 2007, Henry et al., 2008, Huang et al., 2008. LPSs are also able to activate EDO production, leading to increased kynurenine metabolites and tryptophan breakdown. O'Connor et al., 2009, Fu et al., 2010, Dabas et al., 2012. Research into the relationship between LPS, intestinal permeability, and depression has demonstrated that LPSs have the capacity to induce depressive behavior in animals. Yermia, 1996, Henry et al., 2008. Interestingly, antidepressants such as fluoxetine and imipramine attenuate behavioral responses to immune challenges, such as LPS. Yermia et al., 2001. In a recent study, the reverse was demonstrated where LPS reduced the antidepressant effects of fluoxetine in a chronic, unpredictable mild stress model in mice. Wang et al., 2011. Investigations into the incidence of intestinal hyperpermeability in patients with major depressive disorder have confirmed an increased translocation of LPS from gram-negative enterobacteria in depressed patients. This was demonstrated by increased serum IgM and IgA levels directed against LPS of gram-negative enterobacteria. Moss et al., 2008b, 2012b. Moss and colleagues concluded that depressive disorder was accompanied by increased gut permeability, which was associated with an immune response directed against LPS produced by enterobacteria. Effects of curcumin on the LPS-induced immune response. Several studies have investigated the protective effects of curcumin on LPS administration. In two separate studies, curcumin decreased no production in LPS-stimulated microglial cells in a dose-dependent manner. Jung et al., 2006, Tak Aruz et al., 2012. Both pre- and post-treatment with curcumin diminished LPS-induced dopamine neurotoxicity in a dose-dependent manner. LPS-induced production of many pro-inflammatory factors and their gene expressions such as TNF-alpha, NO, PGE2 and IL-1 beta was also dramatically reduced following curcumin treatment. Curcumin also decreased LPS-induced activation of two transcription factors, nuclear factor kappa B and activator protein 1, Huang et al., 2008. The benefits of curcumin may also occur through its ability to reduce the translocation of bacteria into extra-intestinal sites, such as mesenteric lymph nodes, liver, spleen, and/or bloodstream. Carotep et al. 2010. Given the potential of LPS to induce depressive-like behavior in animals, and an increased translocation of LPS in depressed patients resulting from increased intestinal hyperpermeability, ameliorating effects on LPS-induced inflammation provides another potential antidepressant mode of action of curcumin. Conclusion: while clinical trials on the antidepressant effects of curcumin are required to determine the efficacy of this natural substance, this paper highlights the multiple potential antidepressant modes of action of curcumin. As discussed above and summarized in Figure 4, curcumin has been shown through in vitro and in vivo studies to influence a range of neurotransmitters and hormones commonly observed to be disturbed in major depression. It also has the potential to provide neuroprotective effects, and shows some promise as a protective agent against stress-induced neurotoxicity. Curcumin's anti-inflammatory and antioxidant effects are already well recognized and may prove to be another potential mode of action in treating depression. Unfortunately, a commonly cited problem associated with curcumin relates to its poor bioavailability via oral ingestion, limiting its potential clinical application. Anand et al. 2007. However, recent efforts to enhance its bioavailability have shown significant promise. For example, blood levels of the patented formulation BCM95 registered CG was found to be 6.93 times higher than standard curcumin and 6.3-fold higher than a curcumin, lecithin, piperine formula. Antony et al. 2008. In another human study on the patented curcumin formula Mediver registered, its total curcuminoid absorption was shown to be 29-fold higher than an unformulated curcuminoid mixture. Cuomo et al. 2011. Curcumin administered with the agent piperine also increases oral bioavailability. Shoba et al. 1998. While no research has yet assessed whether this increased absorption is associated with increased clinical efficacy, it is hoped that these methods of delivery may at least partly overcome problems associated with curcumin's poor oral absorption and therefore enhance its utility in clinical settings.